Hello, everybody. Welcome to another edition of Global IMMUNOTALKs. My name's Laura McKay. I'm from the Doherty Institute at the University of Melbourne. And it gives me great pleasure today to host this week's Global Immuno speaker. We're coming to you from sunny Australia on this pre-recorded edition. And it's a great privilege to introduce this week's speaker, Dr. Kim Good-Jacobson. But before I introduce our Global Immuno speaker on behalf of all our 2022 Global Immuno organizers, I would like to remind you that, as usual, all the Q&A will be conducted after this talk, and this will be done on Twitter. And I will put up a slide later on following the lecture, which will just remind everybody what hashtags to use and how that works. And also remind you to join us again next week, where we'll have Kiki Fairfax from the University of Utah. So please join us again next week and stay tuned for that. So without further ado, I'd like to introduce this week's Global Immuno speaker, Dr. Kim Good-Jacobson. So Kim is a full professor at Monash University, where she leads the B-cell antibody and memory laboratory. Kim is originally from Australia, where her laboratory now is. She completed her PhD in Sydney at the Centenary and Garvan Institutes before then moving to Yale for a postdoctoral stint. Following returning to Australia, Kim first joined the Walter and Eliza Hall Institute and then started up her laboratory at Monash in 2015. If some of you aren't familiar with some of Kim's work, she's an absolute trailblazer in Australian immunology, one of our brightest young stars. Her research has made incredible contributions on how long-lived antibody-secreting cells and memory B cells are regulated, working in the dysregulation of antibody responses following chronic infection, doing lots of epigenetic and fundamental research into memory B cells and how these cells can be affected by their environment. And Kim's work, rightfully so, has been recognized by a multitude of awards, including the Victorian Young Tall Poppy, and also she is a Bellbury-Vietel Fellow, which is an extremely prestigious fellowship here in Australia. So Kim, we welcome you. We're so happy and privileged to have you join us as a Global Immuno speaker. As you know, one of the traditions here as part of our program is to ask a question to get to know you a little bit better. And so what we'd love to hear from you is if you could go back to your 25-year-old self when you're just starting your career, and you could give yourself a piece of advice that would send you on your way, we'd love to know what that would be. So over to you, Kim. Thank you.
Hi, Laura. So apologies for the swearing, but I think if I could go back and give myself advice, it would be the words of June Osborne from The Handmaid's Tale, which is, don't let the bastards grind you down. So that's pretty much it.
Kim, there's absolutely amazing advice. It's absolutely true. Keep calm and carry on. And I think that's really succinct advice that everybody should take on board early on. Keep going, everyone. Kim, thank you so much. And we can't wait for your talk. If you can share your slides with us, we're excited to hear some of your latest research. Thank you, Kim.
Thanks, Laura. Okay, great. Well, thank you to the organizers for inviting me to give my talk today. So I'll be talking in the first half on some work that we just recently published. And in the second half, I'll be talking about our upcoming story that we're about to submit. So this is pretty much encapsulates what my lab works on. We work on B cells, antibody, and memory B cells. All of you probably do not need this introduction, but I'll give it anyway. So basically, fundamentally, we work on how B cells induce antibody responses during a primary challenge, how we form immune memory, and how that immune memory is able to rapidly differentiate into high affinity antibody-secreting cells upon secondary challenge. But one of the other major questions we ask is, why can't the antibody response be mobilized effectively in chronic infection? And so although we have a number of very successful vaccine stories, we still have a number of challenging pathogens for which we don't have effective vaccines, such as HIV, hepatitis C, CMV, etc. So one of the commonalities is that they induce chronic infection, and this can lead to a dysregulated humoral response. So even though they are very different pathogens and the immune responses can be quite varied, a commonality is that they produce delayed neutralizing antibody, but it's not that they can't produce antibody. They actually have hypergammaglobulinemia, which is too much antibody, and too much immune complexes that become disruptive. So the antibody that's produced is in high amounts and not the right quality. What we also know is that there are changes to the memory B cell population. So the memory B cell population gets remodeled by chronic infection. These changes are quite controversial because there are context-dependent changes that happen to the memory B cells, but that intrigued me to start working on it. So just as a primer on B cell immune responses in terms of effective B cell immune responses, and then what happens when you drive an ineffective B cell response. So in a typical T-dependent response, you have B cells and T cells that are activated by antigen coming together. You can produce early memory B cells in the first week of a response. Some of the other B cells will go on to form a germinal center. And for any non-B cell people out there, this is a training center for B cells to become higher in affinity. So basically, they undergo rounds of somatic mutation, which introduces mutations into the genes encoding the B cell receptor, and then rounds of selection to select higher affinity cells. And so it's like an evolution in miniature. So throughout the response, you'll get production of plasma cells that will become higher in affinity. You'll also produce memory B cells. And once the response has resolved, you are left with these two populations. So long-lived antibody-secreting cells, which home to the bone marrow and continually produce antibody, and then memory B cells, which are quiescent cells that recirculate throughout the body, ready for secondary immune response, where they are able to kind of skip all of that training because they're already trained and produce high affinity plasmablasts within days rather than weeks. So another part of an effective immune response are early generation of short-lived, lower affinity antibody-secreting cells as kind of a first wave of protection. But what happens in an ineffective response is that a lot of the B cells get pushed down this pathway. Hopefully my internet is okay. I can say I'm starting to lag. Production of polyclonal B cells, as well as these atypical or ABCs or aged B cells. They have many names depending on the context. But basically what we know is that you do not get protective long-lived immunity. So the three questions at the heart of my research are: how do we produce effective B cell responses? How do B cells become dysfunctional? And if we know the mechanisms behind these first two questions, can we use that to restore their effectiveness? So how do we tackle that? We ask, how do B cells make fate decisions? So how do B cells remodel themselves to become memory B cells or long-lived antibody-secreting cells in an effective response, or perhaps chronic memory B cells or short-lived antibody-secreting cells? The way we do this is that we look at molecular programs within cells. So we really want to understand this relationship between the microenvironment, how that induces changes in genes within B cells, and then how that leads to changes in function. So a lot of work has been done in the last couple of decades to understand transcription factors, particularly key ones that are involved in, for example, germinal center or plasma cell differentiation. But transcription factors don't act in isolation. So it's really a combinatorial process between these molecular factors as well as epigenetic regulators that are able to induce these large programming shifts in gene expression that's required for a B cell to differentiate. Now, we not only study the genes, so that we not only study the programs within the cells, but also the programmers. And for us in particular, we look at histone modifying complexes. So in short, histone modifiers come in and they can modulate chromatin accessibility either into an open chromatin conformation so that transcriptional machinery can come in and bind to their targets, or into a closed conformation that can repress gene expression. So we've been asking in the lab whether histone modifiers have unique roles in regulating B cell fate and function. And although I won't be talking about these papers today, I just wanted to highlight some of my recent work. So work on DOT1L, which is essential for B cell responses to be initiated, which we published a couple of years ago, as well as some older work that I did looking at the histone modifier MOS. But what I will be going into in detail is this question: does chronic exposure to a pathogen result in epigenetic dysregulation within B cells? So to start to work out how to tackle this, we needed first a model to test how B cell responses do in fact become dysfunctional in chronic infection. So I will say that a lot of the work that's been done in this field has been done with human patients and human peripheral blood. But some of the questions that we wanted to ask really needed an in vivo model. So we turned to the LCMV model, which has been used widely in the T cell field, particularly because we are able to use two different strains of LCMV to induce either an effective immune response or a chronic ineffective immune response. So we also needed to develop a tetramer to track LCMV-specific B cell subsets, because in the past, basically people in the B cell field who use LCMV have relied on an adoptive transfer model, which wouldn't allow us to do the experiments that we wanted to do. So we needed a way to look at B cells in a polyclonal response. And we were fortunate enough that Justin Taylor had developed a tetramer. And we've also had assistance from Stephanie Grah. And then putting this all together, what we've been working on is identifying and characterizing different B cell subsets in acute versus chronic infection, and then using conditional gene deletion models to identify key epigenomic regulators. So in this first part, I'll talk about a paper that we published this year on one of those key regulators. So originally in my lab, these two kind of components were separate. We were working on epigenetics in normal B cell responses and working on chronic infection and how B cells get dysregulated. But the two kind of merged in early work, which we haven't actually published, but basically we found this particular repressive histone modifier, which I won't talk about today. But what we found was that it was absolutely required for germinal center B cell responses when we infected or immunized with acute resolving antigens. So either viral infection, flu, or immunization models. But surprisingly, when we infected these mice with a chronic infection, they were actually still able to form germinal centers. So it seemed like there was something that was uniquely different about these germinal centers in chronic infection that were allowing them to kind of escape this checkpoint. So we wanted to take a step back and ask whether germinal centers in chronic infection were fundamentally different than those in acute. So what we did is we undertook RNA sequencing and ATAC sequencing on germinal center B cells isolated either from our acute or chronic infected mice. And as you can see, what we found was that we had a large change in transcriptional programs. So more genes were being expressed in the chronic infection, and that correlated with increased chromatin accessibility. So of course, we want to know about the programmers. So we started to really look into the data to see whether there were epigenetic modifiers that repress gene expression. Our hypothesis was that these might be dysregulated in chronic infection. So in short, there were multiple clues in the data that pointed us to the polycomb repressive complex 1, the canonical version, and in particular BMI1. So the polycomb repressive complexes are two complexes that work together to compact chromatin and a lot of the time repress gene expression. So we had in our data set increased chromatin accessibility around PRC1 targets and upregulation of PRC1 targets. And we also had this plasma cell gene profile in germinal center B cells, even though we had made sure to sort so that we excluded plasma cell markers. So the GC B cells seem to be prone to or poised to differentiate into plasma cells. And the reason why I say that gave us a clue to BMI1 is because we had actually already been working on BMI1 in the lab. And what we knew already was that BMI1 was required for a subset of antibody-secreting cell formation. So using a BMI1 reporter mouse, we had found that BMI1 is not normally expressed in germinal center B cells in an acute response to immunization. And this fits with data on germinal center B cells in humans, which basically do not express BMI1. Although I will say that we did have this very small fraction that was always there, but on the whole, germinal center B cells didn't express BMI1. But when we gated on plasma cells, they had upregulated BMI1. And again, that fit with human data where we see that plasmablasts and then plasma cells upregulate BMI1, and then it's really highly upregulated in multiple myeloma cells. So when we conditionally deleted BMI1 in mature B cells, what we found was that germinal center B cells were able to form, but BMI1 was really important for plasma cell differentiation in an immunization model. But back to our chronic infection. So what we were seeing is that normally BMI1 is not supposed to be expressed in germinal centers, but they were in chronic infection in our data sets. So we wanted to confirm this by using a number of different means. So first of all, we just isolated germinal center B cells and performed qPCR. And then we used our BMI1 reporter, which is a tamoxifen-inducible reporter. And as you can see, that normally in an MPKLH immunization, you only have a tiny fraction of germinal center B cells expressing BMI1. But in the acute infection, you have slightly more. And then in the chronic infection, you have increased levels of BMI1. Even though we had increased levels in the germinal center B cells, plasma cells always expressed more BMI1, even in the chronic infection. And this is just to show the protein as well. So at day 7, there was not that much expression of BMI1 either in the acute or chronic, but you can see that it is increased by day 14 only in the chronic infection. So what is this consequence? What is the consequence of BMI1 expression in chronic infection? So again, we used our conditional deletion of BMI1 and infected these mice with LCMV docile, which is the chronic version. And to our surprise, when we deleted BMI1 in B cells, we had accelerated clearance of virus. So this is looking at day 28. And then you can see over time, even though at day 7, they have basically very similar amounts of viral load, in the BMI1 deficient mice, they were able to clear it at a faster rate. So in chronic LCMV, splenomegaly is one part of the phenotype of the response. And you can see when we deleted BMI1, we had smaller spleen sizes and a smaller number of splenocytes. Another feature of chronic LCMV is this disruption to B cell and T cell areas, which you can see in the top row of the histology. And again, when we deleted BMI1, the B and T cell areas were restored to what would look like a normal B cell T cell segregation. Okay, so because we're deleting in BMI1 in B cells, and we know that it's important for antibody-secreting cells, we wanted to ask what was happening to the quality and quantity of antibodies, because antibodies, and I mean, we know, everyone knows how important neutralizing antibodies are, but immune complexes, which are antigen antibody complexes, are important for both the adaptive and the innate immune systems. So they're important to drive selection in the germinal center for high affinity variants. And it's also important to modulate the function of innate cells. So when we deleted BMI1 in B cells, what we found is that we had a reduction of antibody-secreting cells, but importantly, we didn't have an absence. So as you can see here, what happened when we deleted BMI1 is actually the chronic infection levels of antibody-secreting cells came down to roughly about what you would find in the acute wild type levels. That was the same for germinal center B cells as well as IgG2C immune complexes. We wanted to double check that this was intrinsic to the B cells. So we set up a bone marrow chimera, a 50-50 chimera. And as you can see, when these chimeras were infected with chronic LCMV, while the Li5.1 still produced a lot of antibody-secreting cells, the BMI1 deficient cells did not. Okay, so that's quantity. What about the quality? So we took our antibody, we took out serum to assess the neutralizing capacity. And what we found was that the BMI1 deficient mice were producing antibodies that had an increased neutralization or neutralizing capacity compared to the controls. We also assessed the non-neutralizing function because even though this isn't as studied as neutralizing capacity, it is very important. And what we found when we looked at glycan profiles of the FC portion of the antibody from these mice is that they had a significant decrease in one particular glycoform. And this has been correlated to differences in ADCC. And as expected, we had increased ADCC from the antibodies when we basically took out antibodies from BMI1 deficient versus wild type mice, and then put them with natural killer cells just from wild type mice. And those NK cells had an increased capacity to induce ADCC. So just to summarize that portion, BMI1 is upregulated during chronic LCMV infection compared to acute in B cells. Deletion of BMI1 accelerated viral clearance and restored splenic architecture. And BMI1 regulates the neutralizing and non-neutralizing function of antibody. So how does it do that? Again, we turn back to our sequencing data to look for clues. And one very red big flag for us was that there was upregulation of MYC targets in the chronic infection. So what we did is we took out tamoxifen-inducible BMI1 reporter mouse and sorted out BMI1 expressing B cells versus BMI1 negative B cells in either the acute or chronic infection. And as you can see, there is a very significant upregulation of BMI1 in germinal center B cells, significant upregulation of MYC in BMI1 expressing germinal center B cells in the chronic infection. So to look into this relationship a little bit further, what we did is we took histology, and I will say one of the major clues for us is that there is this long-standing relationship between BMI1 and MYC in B-cell lymphomas, but hadn't really been addressed in chronic infection before. So we assessed the germinal centers, MYC expression in germinal centers in chronic infection. So these are just in wild-type mice. What had previously been reported in germinal center B cells in acute resolving immunization models is that normally MYC is only supposed to be expressed by a few cells in the germinal center. But you can see in the histology that a lot of cells were lighting up for MYC. When we deleted BMI1, what we found is that we could restore it back to what looked to be kind of more of a normal expression pattern of MYC in those germinal centers. This also correlated to changes within selection, which we assess by B-cell receptor sequencing, but I don't have time to show that today, but it's in the paper. We also wanted to investigate what are the upstream regulators. So what is it in the chronic microenvironment that may be inducing MYC and BMI1 upregulation? It had been previously published that IL-21 is a cytokine that is upregulated in chronic infection compared to acute in LCMV, which we confirmed as well in our model. And so what we did is we stimulated B cells and then added in increasing amounts of IL-21 to see if those high levels of IL-21, and it is really important to focus on the high levels because IL-21 is normally a critical cytokine for B cell responses. So it's not just the presence that may be aberrant because it's a normal component of the B cell response. It's those really high levels. And what we found is that at those high levels, we did get significant increase in BMI1 and MYC. So just to summarize that portion, BMI1 deletion restores MYC expression and positive selection in chronic LCMV infection. We also explored a mechanistic hypothesis of non-canonical versus canonical PRC1, which again, I don't have time to go into, but basically, normally the non-canonical PRC1, which doesn't express BMI1, is expressed in germinal center B cells. And what we think is that in chronic LCMV, it's switching to the BMI1 containing canonical version, which is inducing plasma cell differentiation too early. We also assessed whether BMI1 targeting can reduce steady state antibody-secreting cells, because obviously in a disease like lupus, that is one of the things that people are chasing afterwards. And we found that we could. But lastly, for this part of the talk, we asked whether BMI1 could be targeted with a small molecule inhibitor to reduce detrimental antibody-secreting cells and immune complexes. So first, what we did is we stimulated B cells in vitro to induce plasma cell differentiation and cultured with increasing levels of a small molecule inhibitor to BMI1. And as you can see in the box, we had selective decrease in antibody-secreting cells with increasing concentrations. We then looked in vivo. So we infected mice and then administered a small molecule inhibitor daily and then assessed at 14 days post-infection. And what was noticeable almost straight away is that normally with chronic LCMV infection, you have a continual loss of body weight. And what we found is when we administered the small molecule inhibitor, the mice were able to start recovering their body weight much earlier. And then when we assessed at day 14, we found again that we had a significant decrease in both antibody-secreting cells and immune complexes. So just to summarize this part of the talk, what we found was that BMI1 is a crucial immune modulator that controls effective antibody-mediated responses to viral infection, which is so critical for both the adaptive and innate immune responses. So what we think is that BMI1 is kind of, falls into this Goldilocks principle where you do want BMI1 as part of your normal response to produce plasma cells, or at least a subset of plasma cells. But in chronic infection, when the B cells are trying to adapt to high levels of inflammation, high loads of virus, that the B cells are using this mechanism to push out more antibody-secreting cells, but before that they're ready. And so that then you induce this kind of ineffective antibody response. And again, this was published earlier this year. Okay, so at the start, I talked about memory B cells quite a bit, but I haven't talked about them yet. So what about memory? So memory B cells have actually I've always tried to focus on memory, but every time I start off with a hypothesis and a mouse, it always leads me down an antibody-secreting cell phenotype. But I am always trying to come back to memory B cells. So when I did my PhD a long time ago, I really wanted to tackle this question, which is, we know that memory B cells have an intrinsic advantage to succeed. So work that I did during my PhD showed that you could basically reduce down stimuli to almost nothing, and the memory B cells would always enter division earlier than naive B cells. And then work I did in my second postdoc at WeHi showed that if you could modulate, when you modulated the epigenetic regulator MOS, you can modulate different memory B cell subsets and their function. So we also know that immune memory is a diverse population in both health and disease, and that diversity enables dichotomous functions, but particularly the ability to persist for long periods of time, but also to differentiate into high-affinity plasmablasts in secondary reactions. But besides that, we don't really know how the microenvironment modulates this diversity within the memory population during an initial response, and particularly how chronic infection remodels that memory B cell diversity. So to understand this, this is mainly work from a very talented PhD student, now postdoc, Lucy Cooper. What we did is we used all those tools at the start to establish an in vivo chronic memory model. She did, I won't show most of that work, but she spent a lot of years actually setting that up. And then we got to a point where we could do single cell sequencing. So we performed single cell RNA sequencing, single cell BDJ sequencing, which I won't show today, and single cell ATAC sequencing to really be able to investigate the fundamental epigenomic changes that happen in memory B cells during chronic infection. So what we found, and I'll come back to this data, is that we did have diversity within the memory B cell population, which we expect, but it's really modulation of one dominating acute cluster in both ATAC and RNA-seq that we would lose in the chronic infection, and one that would emerge that wasn't there in the acute. Okay, so for the first part, what she did was she established and characterized an in vivo chronic memory B cell model. And just to kind of summarize all of the work that she did, what we found was that there are a range of changes that are in line with human data, such as an increase in CD11C positive, CD21 low memory B cell subset, which you see in HIV, malaria, and other forms of chronic infection. And then with PD-L2 and CD80, which work that was done in Mark Slomchik's lab showed could segregate memory B cell subsets. What we found was that there was a shift towards double negative cells in the chronic infection, which are the more naive like memory B cells. And then we also saw an increase in CD21 low memory B cells in the peripheral blood, which again is what happens in patients where you see this emergence of this population dominating in the peripheral blood. Okay, so as I mentioned, single cell RNA-seq identified key memory B cell clusters that were either expanded or diminished in chronic LCMV infection. And so basically, I did take off all the gene names because this is going up on YouTube and we haven't published it yet, but we're going to submit it soon. So stay tuned. But basically what we found is that the subset that was present in acute but diminished in chronic was more of a BCR signaling subset. Conversely, the one that seemed to emerge in the chronic infection was dominated by ISGs. I also wanted to point out that there was a different cluster, cluster 5, and apologies, I realize I've taken off the cluster numbers, but this cluster here in the blue, that's present in both the acute and chronic, and that is the Tibet related cluster, which people tend to work on in this field. So we had a number of gene sets that were enriched in the chronic infection, including interferon signaling. And what Lucy did is she compared these to other data sets. And so data sets looking at lupus, which has a similar phenotype, as well as hepatitis C or postmortem lung tissue of COVID-19 patients were enriched in our chronic infection. So that was to kind of set up what are the changes in the genes that are being expressed in chronic infection, or the diversity within the memory B cells at the gene expression level. But what we really wanted to do is to identify whether there were particular epigenomic changes between memory B cells generated in resolving infection versus chronic viral infection. And so we used single cell ATAC sequencing to do that. And then based on this, what we wanted to do is to follow on and ask what drives these changes in chronic viral infection, and whether those chronic memory B cells are adaptable or whether those changes are locked in. So as I mentioned, what we did is we took out tetramer-specific B cells and undertook single-cell ATAC sequencing. What we found was that there were 7 unique clusters. And again, basically, there was one major cluster that was present in acute and diminished in chronic, and then vice versa. So you can see that the red is the one that diminishes in the chronic, and the orange is the one that expands. We looked at where the peaks were sitting, and we didn't see any differences between acute and chronic in terms of their genomic location. So then we wanted to look into these specific or these unique clusters in more detail. One of the questions in the field is whether those cells that emerge in the peripheral blood are there already, but just are expanded or maybe have different migration properties in chronic infection. What we did, and I should say there are some important studies in malaria and flu showing that you can generate them, for example, in flu. What we wanted to do is ask for these 2 kind of unique clusters, we can find a few of those cells in the corresponding data set. We wanted to ask whether they were actually the same or whether they were quite different. So we compared them, and there wasn't actually that many changes that we could find when we compared the dominating cluster in either acute or chronic to its corresponding cells in the other infection, suggesting that they are actually produced in the other infection. They're just very, very few. We also looked at cluster 5, which is kind of sitting away from everything else. And what I'll show you is that we know that they're germinal center B cells. But we wanted to look to see whether we find differences that we found in our paper that we published earlier this year, even though these are 2 weeks later in the infection. So these are 4 weeks post-infection, whereas the data set I showed you before is 2 weeks post-infection. And we did find a number of differentially accessible regions in both data sets. So just to back up what I just said about the germinal center B cells. So what we show in the single cell RNA-seq, as well as the single cell ATAC-seq, is the germinal center B cells kind of sit away from everything else. And you can see here, for example, A5, which is that germinal center cluster, has upregulated AICDA, which is the gene that encodes AID, and downregulated cell, which is the gene that encodes CD62L. Now, what we wanted to do is to look at how related each of these clusters are. And again, as you can see, A5, which is the germinal center cluster, is kind of sitting away from everything else, as we would expect. But then when we kind of hone in on the non-GC clusters, what we find is A1, which is the chronic dominant cluster, and A3, which is the acute dominant cluster, actually sitting on 2 very different branches. A6 here, which is this pink cluster, is again that TBET related cluster. So one of the things we were kind of wondering is whether they were very similarly related, these 2 clusters that change between acute and chronic, but they seem to be quite different. So what we've been doing is to understand the chromatin changes the unique chromatin changes in this chronically expanded cluster in much more detail. So obviously we've looked at what those significant DARs are in cluster 1 compared to the other non-GC clusters. And it was the most unique compared to the rest of them. And cluster 6, which was that TBET and ZEB2 cluster, was the second most unique. So even though it was there in both acute and chronic, it did have a range of differentially accessible regions compared to the other non-GC B cells. I am trying very hard not to say memory B cells too much, because some of those cells, there'll be a few naive B cells. And then it's a little bit of semantics as to whether calling the rest of the memory B cells, where in chronic infection, because you have an ongoing infection, they are cycling still, or at least a portion of them are. But anyway, so that from that data set, what we've done is we've identified transcription factor motifs that are, uniquely or differentially accessible in these different data sets as well. And again, cluster 1 was more unique, but I won't be showing that data today. So just on to the final part, which is using this data set or setting up, understanding how memory B cells are epigenomically imprinted from their microenvironment. So what we really wanted to ask is how persisting high viremia and inflammatory signals in chronic infection, how they remodel memory B cell subsets, when and which ones are most important, and whether those memory B cell subsets are adaptable to change later in the response. And there's quite a few studies in the HIV field that show changes to antiretroviral therapy, basically that the patients, their memory B cells rebound very quickly, depending on the timing. And so we really wanted to assess that in much more detail in our model. So what we did is we used the antiviral favipiravir to clear LCMV at different time periods, early in the response or later in the response. We changed the initial dose of the LCMV docile because you need to induce the response with a higher dose than you're giving of LCMV-we, so we changed to a lower dose. And then we looked at interferon blockade at different time periods as well. So in short, when we used favipiravir, when we first looked at it in kind of the later part of the response, so after 4 weeks post-infection, we saw that we could accelerate viral clearance when we gave favipiravir. But what we found was that favipiravir did not modulate memory B cell phenotype. We changed the dosing schedule a lot, but we couldn't find one that significantly altered these populations. Similarly, when we induced infection with an initially lower dose of LCMV docile, it didn't significantly alter these populations at day 28 either. But as I mentioned before, we had this interferon-driven ISG signature that was enriched in chronic infection, which is a little bit surprising because we know that in acute infection, you actually get these really high levels of interferon early in the response. But what we do know as well is that you have this early robust interferon is very important for driving an effective response. But when you have delayed but kind of persisting levels of interferon, that can drive severe disease. And there was a great review published a couple of years ago. So what we found was very similar, that you have this high robust interferon, and a lot of people have looked at this in LCMV Armstrong and Clone 13, and we see similar things in lean docile, that acute induces high levels early, but then shuts off basically, whereas the chronic takes time to induce interferon and then persists at a similar level. So even though at day 4 it looks like it goes down, it's actually at the same level as it is at day 30. So can we reprogram memory B cells by therapeutically targeting interferon signaling? So what we did is we used blocking antibodies to ask these questions. Are chronic memory B cells maintained if we block interferon late in the infection? And how is the chromatin architecture of memory B cells modulated by interferon signaling? So we used either an anti-interferon gamma or an anti-IFNA1 antibody to assess the populations by flow cytometry or by bulk ATAC sequencing. So the reason why we did early and late is because we initially looked at the late response, but didn't seem to find many differences when we blocked interferon. And what had been published by Mark Schlumschick in 2016 was that memory B cells tend to arise earlier in the response. And what we see is the same when we do BRDU pulse chase labeling experiments. When we pulse early in the response, the acute memory B cells are maintained for long periods of time. The chronic aren't maintained to the same level, but they have a faster decay rate, I guess you would call it, because they are still cycling to a greater extent in the chronic infection. Whereas when you pulse later at 2 weeks or 4 weeks post-infection, even though you are producing memory B cells, they tend to lose the BRDU. So we blocked either early in the response or late. And what we found is when we blocked late in the response, we didn't see any phenotypic changes. But when we blocked early in the response, we found an expansion of overall B cell numbers and a shift in the memory B cell phenotype. And so this is just one example where we see a shift back in those PD-L2 CD80 populations, where normally in the chronic infection, you get more double negatives than acute, but we tend to see it shift to increasing in PD-L2 CD80 double positives when we block IFNA. So it looks like early was more important, even though we are producing memory B cells throughout the response, particularly in chronic infection where they're continually cycling. So we wanted to ask, do we see those changes at the chromatin level? Can the chromatin architecture be modulated by differences in interferon signaling? And this is kind of testing our hypothesis that there's a hit and run model of epigenomic imprinting within memory B cells in the early part of the response. And what we did see is that the early part of the response was really where memory B cells could be remodeled by blocking type I interferon. So in the early blocking phase, what we found was that there were over 600 differentially accessible regions when we blocked IFNA. So you can see that there's a portion that downregulate or decrease accessibility and a portion that increases accessibility when we block IFNA. We see some changes in the acute infection, but basically we see the decreased accessibility, but we don't see that corresponding increased accessibility. Compared to when we look late, so when we block late in the response or later in the response where the acute has already cleared the virus, but the chronic virus is still present, what we found is that we didn't see that many changes, either when we blocked with interferon gamma or IFNA blocking antibodies. So just to summarize that part of the talk, what we were asking was what drives epigenomic changes in memory B cells during viral infection, and whether chronic memory B cells are adaptable or whether those changes are locked in. And what we found was that, out of all of the things that we tried, which is quite a lot, it was really blocking IFNA in the early part of the response in chronic infection. So it's not from the very start. We delayed the onset of the antibody to allow that first couple of days of the response. But we were able to modulate memory B cell phenotype as well as the memory B cell epigenome. And so I wanted to leave you with this, that epigenetic regulators and basically how they set up the memory B cell population and drive antibody-secreting cell differentiation is really critical to balance what you want in a normal response and how the B cells are able to adapt to the microenvironment and how that potentially goes wrong in chronic infection or chronic inflammation. And so I will finish by thanking everyone involved. The first part of the talk, Andrea Di Pietro was a postdoc in my lab who really drove the BMI1 story. Lucy Cooper really drove the second part of the work. And I'd like to thank our collaborators at Monash in Australia and internationally, and our funding bodies. And I will stop there. Thank you.
Thank you so much, Kim. That was such a huge body of work and such a treat for you as well to share some of your unpublished work with us. Thank you so much. And so I'm sure everybody has so many questions for Kim. And so with that, I will share my screen again and let everybody know how to ask Kim those questions. So please get onto Twitter. Of course, we're pre-recording in Australia right now, but Kim has kindly agreed to jump onto Twitter and answer your questions in your own time zone after the talk. And so please search for the account Global IMMUNOTALKS, find the tweet that says, ask for questions for Dr. Kim Good-Jacobson here, and then reply to that tweet with our hashtag, and Kim will be answering from her own Twitter account. So that just leaves me to remind everybody to tune in next week. We'll have Dr. Kiki Fairfax from the University of Utah to give Kim such a huge thank you again. Thank you so much for the time it's taken you today to share your beautiful data with us. And that leaves me to close the session. Thanks everybody for watching, and we will see you next time. Thank you so much.
Thank you.